More than 470 million years ago, Earth looked nothing like the green world we know today. There were no forests, no flowers, and no grassy fields. Most land was bare rock, with only water covering much of the planet. Then tiny early plants began one of the greatest changes in Earth's history. They slowly spread across the land, changed the air, helped create soil, and made life on land possible for countless other living things. Earth was a very different place long before the first plants appeared. There were no forests, no grasslands, no flowers, no moss, and no trees. There was no green colour spreading across the land because nothing had yet developed that could grow on dry ground. The surface of the planet was made up of bare rock, flowing water, thick clouds, active volcanoes, and empty land. Every part of the world was shaped by natural forces that worked without any plant life. Wind moved across open ground without passing through leaves or branches. Rain fell onto stone instead of soil. Rivers carried pieces of broken rock into the oceans. Waves struck empty shorelines that had never supported roots or stems. Earth itself formed about 4,540,000,000 years ago, from a large cloud of dust and gas that surrounded the young sun. Tiny pieces of rock and metal moved around the sun and slowly crashed into one another. These collisions became larger over time. Heat from these impacts, along with heat from materials deep inside the growing planet, caused much of the early Earth to melt. The surface became an ocean of hot, liquid rock. Heavy materials such as iron and nickel moved toward the center, forming Earth's core. Lighter materials stayed closer to the surface and slowly became the mantle and the outer crust. During these early times, the young planet experienced constant impacts from large space rocks. Some of these objects were small, while others were many miles across. Each impact released a huge amount of energy. The force melted rock, created giant craters, and threw hot material high into the sky. Some impacts changed large parts of the planet's surface. Others may have helped bring water and other important materials that later became part of Earth's oceans and atmosphere. The young Earth was much hotter than it is today. Heat escaped from deep inside the planet through countless volcanoes. Thick streams of melted rock spread across the surface. Many volcanoes released large amounts of gas into the air. These gases included water vapor, carbon dioxide, nitrogen, sulfur gases, and other materials. The early atmosphere looked nothing like the one people breathe today. It contained almost no free oxygen. Any oxygen that was produced quickly reacted with rocks and minerals instead of remaining in the air. The sky may have looked very different because of the gases and particles floating in the atmosphere. Thick clouds often covered large areas. Ash from volcanic eruptions filled the air after major eruptions. Dust from impacts also remained in the atmosphere for long periods. Sunlight sometimes reached the surface clearly, while at other times it was partly blocked by clouds and volcanic material. As the planet slowly cooled, the outer layer hardened into solid rock. This first crust was not stable. Heat rising from below caused it to crack, break apart, and melt again in many places. New rock formed while older rock disappeared beneath the surface. This movement continued for millions of years. The crust slowly became stronger, but it remained active because the inside of Earth was still extremely hot. Water played a major part in changing the planet. Much of the water came from gases released by volcanoes. As the atmosphere cooled, water vapor changed into liquid. Rain began to fall. This was not a short period of rain. Scientists think rain may have continued for very long periods as the atmosphere slowly lost its water vapor. The falling water collected in low areas, creating the first oceans. More water may also have arrived from icy objects that struck Earth during its early history. The first oceans were different from those of today. Their chemistry changed over time as water mixed with minerals from rocks and gases from volcanoes. Rivers carried dissolved materials into the sea. Underwater volcanic activity added more chemicals. The oceans became places where many natural chemical reactions took place. These reactions slowly changed the makeup of the water over millions of years. The land remained completely empty of plants. There was no soil covering the rocks because soil depends on many living things. Today, plant roots, fungi, bacteria, insects, worms, and other organisms help create soil. Before plants existed, these living systems had not yet developed on land. Instead, the ground consisted mainly of exposed rock, loose stones, volcanic material, sand created by weathering, and broken pieces of minerals. Weather slowly wore down the rocks. Rain entered small cracks. Changes in temperature caused rocks to expand and contract. Flowing water carried away tiny pieces. Wind moved dust and sand across open ground. These natural processes slowly broke large rocks into smaller pieces. Even without plants, weather continued to shape mountains, valleys, and coastlines every day. Volcanoes were among the most powerful forces on the planet. Some erupted beneath the oceans, others rose above the surface. Lava flowed across large areas before cooling into solid rock. Ash covered nearby land. Gases escaped into the atmosphere. These eruptions helped create new land while destroying older landscapes. Volcanic islands appeared in some places. Other islands disappeared as they sank or were covered by later eruptions. Earth's continents looked nothing like the ones seen today. The crust moved slowly because of activity deep inside the planet. Pieces of crust separated, collided, and changed position over millions of years. Small landmasses formed and later joined together. Some broke apart again. The shapes of coastlines changed many times. Oceans opened and closed as the crust continued to move. The rocks found on the early surface were mainly volcanic. Many formed when lava cooled after reaching the surface. Others formed deep underground before later being lifted upward. Heat and pressure changed many rocks into new forms. These changes continued as Earth's interior remained active. Layers of rock recorded parts of the planet's long history, although many of the oldest rocks were later destroyed by melting and movement. Without plants, the land had almost no protection from rain and wind. Water flowed freely over bare rock. During heavy rainfall, large amounts of loose material moved into rivers. Floods carried stones, sand, and mud toward the oceans. River channels changed direction over time as flowing water cut through the landscape. Some valleys became deeper while others filled with sediment. The shoreline between land and sea was always changing. Waves struck exposed rock day and night. Pieces of stone broke away and became smaller over time. Sand gathered along some coasts while other areas remained rocky. Tides moved water in and out without passing through marshes, grasses, or coastal forests because none of these environments yet existed. The atmosphere slowly changed through natural processes. Volcanoes continued releasing gases. Some gases escaped into space. Others became trapped by Earth's gravity. Water moved between the oceans, atmosphere, and land through evaporation and rainfall. Carbon dioxide dissolved into seawater. Minerals trapped some carbon inside rocks. These slow changes continued over extremely long periods. Lightning flashed through storms above the young planet. Strong winds crossed empty land. Rain fell onto volcanic plains and rocky hills. Rivers carved paths through stone. Waves rolled across wide oceans. Deep below the surface, hot rock continued moving slowly. Earth remained an active world where heat from the interior constantly reshaped the surface. Many minerals formed as rocks cooled or reacted with water. Quartz, feldspar, mica, and many other minerals became common parts of the crust. Iron-rich minerals reacted with the small amounts of oxygen that appeared through natural chemical reactions. These reactions created new mineral forms that remained locked inside rocks. Over time, countless chemical changes occurred without the help of plants. The oceans covered most of the planet. Their floors contained long volcanic ridges where new crust formed. In other places, older crust slowly moved downward into the mantle. Hot water flowed through cracks in the ocean floor. This water carried dissolved minerals and later released them back into the sea. These underwater systems became important places for chemical activity during Earth's early history. Earth's rotation controlled the pattern of day and night. The young moon orbited much closer to Earth than it does today. Because of this, ocean tides may have been stronger than they are now. Water moved across coastlines with great force. These tides shaped shorelines and carried minerals between land and sea. The pull of the moon became an important part of Earth's natural systems. The sun was slightly less bright than it is today, yet Earth remained warm enough for liquid water because greenhouse gases trapped heat in the atmosphere. Carbon dioxide and other gases prevented too much heat from escaping into space. This natural process helped keep the oceans from freezing completely, even though the young sun produced less energy than it does now. The planet experienced long periods of change. Some regions cooled while others remained hot because of volcanic activity. New mountains rose as pieces of crust pushed against one another. Older mountains slowly wore away under the action of wind and rain. Sediment gathered in low areas, where it formed new layers of rock after long periods of pressure. No forests softened the landscape. No roots held the ground together. No leaves slowed falling rain. Every storm acted directly on bare stone and loose rock. The land changed through natural forces alone. Water, wind, heat, cold and gravity shaped every hill, valley, shoreline and mountain across the planet. The surface may have appeared lifeless to anyone standing on the land. Wide stretches of exposed rock extended towards distant mountains. Volcanoes released smoke and ash into the sky. Rivers flowed across empty ground before reaching the sea. Waves broke against silent coastlines. Clouds drifted overhead while rain continued to wear away the surface. Beneath this barren landscape, Earth continued changing from the inside as heat slowly escaped through the crust, preparing the planet for the many natural changes that would unfold over the immense stretches of time that followed. The earliest evidence of life in the ancient oceans takes us back to a time when the Earth was still very young. The planet was very different from the world we know today. There were no forests, no animals, no birds, no insects, and no flowers. The land was mostly bare rock shaped by constant volcanic activity. The skies were filled with gases released by volcanoes, and the oceans were still forming from water that had cooled and collected over a very long period. Even though the surface looked harsh and empty, the oceans slowly became places where the first known forms of life appeared. Scientists believed that life began in water because the oceans provided conditions that could protect simple living material from many dangers found on the land. Water helped keep temperatures more stable. It also allowed chemicals to mix together more easily. These chemicals were the building blocks needed for life. Although scientists are still studying exactly how life first appeared, many pieces of evidence found in ancient rocks show that living organisms existed very early in Earth's history. The oldest signs of life do not come from bones or shells because such things did not yet exist. Instead, they come from tiny chemical traces, microscopic fossils, and unusual rock formations that were created by simple living organisms. Instead, they come from tiny chemical traces, microscopic fossils, and unusual rock formations that were created by simple living organisms. These clues are very small and often difficult to study. Scientists use powerful microscopes and advanced laboratory equipment to examine them carefully. Every new discovery helps researchers understand more about the earliest chapters of life on Earth. The oldest rocks on Earth are very rare because most ancient rocks have been changed, broken apart, buried, or melted deep inside the planet over billions of years. The Earth's crust is always moving because of plate tectonics. Continents slowly shift, mountains rise, and old rocks sink into the planet's interior. Because of these changes, finding rocks that have survived from the earliest part of Earth's history is very difficult. Some of the oldest rocks are found in places such as Western Australia, Greenland, Canada, and parts of southern Africa. These rocks have remained stable for an extremely long time and contain valuable information about the ancient Earth. Scientists carefully collect samples from these locations and study them to look for signs that living organisms once existed. One important kind of evidence comes from tiny amounts of carbon trapped inside ancient rocks. Carbon is one of the basic elements that all known life depends on. Living organisms process carbon in ways that leave behind special chemical patterns. When scientists examine very old rocks, they sometimes find carbon with these patterns. This suggests that living organisms may have been present when those rocks were formed. One famous example comes from rocks in western Greenland that are more than 3,700,000,000 years old. Scientists studying these rocks found carbon that appeared to have been processed by living organisms. This discovery attracted worldwide attention because it suggested that life may have existed much earlier than many researchers had once believed. However, some scientists questioned the findings and argued that natural processes without life could also produce similar chemical patterns. This debate continues as researchers gather more evidence. Scientific progress often happens through careful discussion and repeated testing. When one team announces an exciting discovery, other scientists examine the same evidence. They test different explanations and search for new samples. This careful process helps reduce mistakes and improves our understanding of the past. As more discoveries are made, ideas become stronger or are changed when better evidence becomes available. Another important source of evidence comes from ancient rock structures called stromatolites. Stromatolites are layered rock formations created by groups of tiny living organisms, especially microorganisms known as cyanobacteria. These tiny organisms grow in shallow water where sunlight reaches the surface. As they grow, they trap small grains of sand and other particles. Layer after layer builds up over long periods, creating rounded or dome-shaped structures that slowly become rock. Some of the oldest known stromatolites are found in Western Australia and are more than 3,400,000,000 years old. These ancient structures show that simple communities of microorganisms were already living in shallow seas. The organisms themselves were extremely small, but together they created large rock formations that survived for billions of years. Scientists study modern stromatolites that still grow in a few places today. These living examples help researchers understand how ancient stromatolites formed. Modern stromatolites can be found in places where conditions prevent many larger animals from disturbing them. By watching these living communities grow, scientists gain valuable information that helps them interpret ancient rocks. Cyanobacteria played an important role in the history of life. These tiny organisms were among the earliest forms of life able to use sunlight to produce food. This process is called photosynthesis. During photosynthesis, cyanobacteria use sunlight, water, and carbon dioxide to make food while releasing oxygen into the surrounding water. At first, the oxygen produced by these microorganisms did not build up in the atmosphere because it quickly reacted with iron and other materials dissolved in the oceans. Large amounts of dissolved iron combined with oxygen to form iron minerals that settle to the seafloor. Over countless generations, these minerals formed thick layers of rock, known today as banded iron formations. These banded iron formations provide another important record of Earth's early history. They show that oxygen was slowly being produced by living organisms long before the atmosphere became rich in oxygen. Scientists have found these rocks in many parts of the world, and they help explain how the Earth's environment slowly changed because of life itself. Before oxygen became common, the atmosphere was very different from today's air. There was little or no free oxygen available for breathing. Instead, gases such as carbon dioxide, methane, nitrogen, water vapor, and gases released by volcanoes filled the atmosphere. The first living organisms did not need oxygen. They survived using other chemical reactions that allowed them to obtain energy. Many scientists believe the earliest organisms were extremely simple, single-celled life forms. A single cell is the smallest unit of life, capable of carrying out all the activities needed for survival. These early cells had no nucleus and no complex internal structures like those found in many modern cells. Organisms with this simple structure are known as prokaryotes. Modern bacteria and archaea belong to this group of simple organisms. Although today's bacteria and archaea have changed through evolution over billions of years, they still provide valuable clues about what early life may have been like. Many of these organisms can survive in conditions that would kill most other forms of life. Some bacteria live near deep-sea hydrothermal vents, where hot water rich in minerals rises from cracks in the ocean floor. Temperatures near these vents can become extremely high, yet certain microorganisms thrive there. Instead of using sunlight, they obtain energy from chemicals released by the Earth's interior. This process is known as chemosynthesis. The discovery of organisms living around hydrothermal vents changed many scientific ideas about where life could survive. It also raised the possibility that similar environments may have provided suitable conditions for the earliest life on Earth. Some researchers believe the first living cells may have formed near these underwater volcanic systems, where energy and useful chemicals were constantly available. Other scientists suggest that life may have started in shallow pools, coastal regions, or other watery environments exposed to changing conditions. These places could have allowed important chemical reactions to occur as water levels rose and fell. Different scientific ideas continue to be tested because the exact place where life first appeared remains unknown. The oceans of the ancient Earth were very different from modern oceans. They contained large amounts of dissolved minerals released by volcanic activity and weathering of rocks. The water may have appeared darker because the atmosphere and oceans contained many substances that are much less common today. Even so, these oceans became the setting where life slowly developed. Lightning, volcanic heat, ultraviolet radiation from the sun, and chemical reactions all supplied energy that may have helped simple molecules combine into more complex ones. Scientists have performed laboratory experiments showing that some building blocks of life can form naturally under conditions similar to those believed to exist on the early Earth. These experiments do not create living cells, but they demonstrate that important organic molecules can form without living organisms already being present. Organic molecules are chemicals that contain carbon and are necessary for life. These include amino acids, sugars, fats, and the building blocks of genetic material. Finding ways that these molecules could form naturally helps scientists understand how life may have begun from non-living matter. One famous laboratory experiment performed during the 1950s showed that amino acids could form when simple gases were exposed to electrical sparks that represented lightning. Later experiments using different conditions produced many other important organic compounds. These studies continue today as researchers investigate many possible pathways leading towards the first living cells. Scientists also search for microscopic fossils preserved inside ancient rocks. These fossils are much smaller than grains of sand and require powerful microscopes to study. Some appear as tiny round or thread-like structures that resemble living microorganisms. Identifying true fossils is difficult because many natural mineral structures can look similar. Researchers carefully examine the shape, chemical composition, and surrounding rock before deciding whether a structure is truly biological. This careful approach helps prevent mistakes and strengthens confidence in important discoveries. Some ancient rocks from Western Australia contain microscopic structures that many scientists interpret as fossilized microorganisms more than 3,400 million years old. These discoveries have been studied for many years. While many researchers support their biological origin, others continue to examine whether non-living processes could have produced similar shapes. Scientific debates like these are common in studies of the earliest life because the evidence is extremely old and often altered by heat and pressure. Ancient rocks rarely remain exactly as they were when first formed. Geological processes may change their appearance over immense periods of time. Scientists therefore use many different methods instead of relying on only one type of evidence. Chemical studies provide another important source of information. Living organisms often prefer certain forms of elements over others during their normal activities. These preferences leave chemical signatures that can remain preserved inside rocks. Researchers measure these signatures with highly sensitive instruments capable of detecting tiny differences that are invisible to the human eye. Sulfur, carbon, nitrogen, and other elements all provide useful information. Together, these chemical clues help scientists decide whether ancient rocks were influenced by living organisms or by ordinary geological processes. DNA cannot survive for billions of years, so scientists cannot recover genetic material from the earliest organisms. Instead, they compare the DNA of living organisms today. By studying similarities and differences among bacteria, archaea, plants, animals, fungi, and other forms of life, researchers reconstruct the history of evolution. These studies suggest that all living organisms share a common ancestor that lived very early in Earth's history. This ancient ancestor is often called the last universal common ancestor. It was not the first living organism, but rather the ancestor from which all living organisms alive today are believed to have descended. The last universal common ancestor was probably a simple, single-celled organism. It likely possessed genetic material, proteins, cell membranes, and the ability to reproduce. Scientists continue to study modern microorganisms to better understand what this ancient ancestor may have been like. Life during these early times remained microscopic for an extremely long period. There were no fish, plants, insects or animals swimming through the oceans. Instead, the seas contained countless tiny cells floating freely or forming thin layers on rocks and sediments. Although individually invisible to the naked eye, their combined activity slowly changed the chemistry of the oceans and atmosphere. As microorganisms multiplied, they spread into many different environments. Some lived near coastlines, others occupied deeper water. Some survived near underwater volcanoes, while others settled on muddy seafloors. Different groups developed different ways of obtaining energy and nutrients. Some early microorganisms fed on chemicals dissolved in the water. Others consumed substances produced by neighboring organisms. Some learned to use sunlight through photosynthesis. This variety allowed life to expand into many parts of the ancient oceans. The appearance of photosynthesis had enormous effects on the planet. Oxygen slowly accumulated in seawater before eventually entering the atmosphere. This process happened over an extremely long span of time. At first, oxygen reacted with iron and other substances, preventing it from building up freely. Eventually, these materials became less abundant, allowing oxygen levels to rise. The increase in atmospheric oxygen transformed Earth's environment. Organisms that could use oxygen gained access to a powerful new source of energy. This eventually supported the evolution of larger and more complex forms of life. However, this change occurred long after the earliest life first appeared. Many ancient microorganisms probably lived in large communities called microbial mats. These mats formed thick layers covering rocks and sediments in shallow water. Different kinds of microorganisms occupied different layers depending on the amount of sunlight, oxygen, and chemicals available. Each layer carried out its own activities while interacting with neighboring organisms. Modern microbial mats, found in certain coastal areas, provide valuable examples of how ancient communities may have functioned. Scientists study these living systems to understand nutrient cycling, photosynthesis, chemical reactions, and interactions among different microorganisms. Ancient oceans also experienced frequent volcanic eruptions beneath the water. These eruptions released heat, gases, and minerals that influenced ocean chemistry. Hydrothermal systems formed where seawater entered cracks in the ocean floor, became heated deep underground, and returned carrying dissolved minerals. These environments supported unique microbial communities. Meteorite impacts also affected the young Earth. Large space rocks struck the planet more often than they do today. Some impacts caused enormous explosions that heated the atmosphere and oceans. Despite these dangerous events, evidence suggests that life survived and continued evolving. Simple microorganisms may have been able to recover more easily from global disasters than larger organisms would have. Some scientists have even suggested that organic molecules from space contributed to the ingredients needed for life. Meteorites contain amino acids and other carbon-rich compounds. While these materials alone do not prove that life came from space, they show that important building blocks can form beyond Earth and reach our planet naturally. The study of ancient life combines many branches of science. Geologists examine rocks, chemists analyze ancient molecules, biologists study living organisms, paleontologists search for fossils, physicists help develop instruments capable of detecting tiny chemical signals. Together, these experts build a clearer picture of Earth's earliest history. Dating ancient rocks is another important part of this work. Scientists often use radioactive elements trapped inside minerals. Certain elements slowly change into different elements at steady rates over immense periods of time. By measuring these changes, researchers estimate the age of rocks with remarkable accuracy. This dating allows scientists to place discoveries into the correct order. They can determine whether certain fossils, chemical signatures, or rock formations appeared before or after important environmental changes. Accurate dating helps create a detailed timeline for the history of life. Even with advanced technology, many questions remain unanswered. Scientists continue searching for older rocks, better preserved fossils, and clearer chemical evidence. Every new discovery has the potential to improve our understanding or challenge earlier ideas. This ongoing process is one of the strengths of scientific research. The earliest oceans covered much of the Earth's surface and provided countless environments where simple life could survive. Shallow coastal waters received sunlight needed for photosynthesis. Deep ocean regions contained chemical energy released from the Earth's interior. Sediments trapped organic material, allowing some evidence to survive for billions of years. Tiny microorganisms reproduce by dividing into new cells. Over many generations, small changes accumulated through evolution. Some changes improved survival under certain conditions. Organisms with useful traits became more common over time. This gradual process allowed life to become increasingly diverse. Although early life remained simple for a very long period, it laid the foundation for everything that followed. Every plant, animal, fungus, and microorganism living today traces its history back to these ancient, single-celled ancestors that lived in the oceans billions of years ago. The earliest living cells also interacted continuously with their environment. They absorbed nutrients from surrounding water, released waste products, and influenced chemical cycles throughout the oceans. These activities slowly reshaped the planet in ways that later allowed increasingly complex ecosystems to develop. Long before animals, forests, or flowering plants appeared on Earth, tiny living organisms were already shaping the planet in ways that would affect every form of life that came later. Among the most important of these early organisms were photosynthetic microbes. These microscopic living things were able to use sunlight as a source of energy. They lived mainly in shallow seas, along coastlines, and in other places where sunlight could reach the water. Although each individual microbe was extremely small, their combined activity over an enormous span of time changed the air, the oceans, and the surface of the planet. The early Earth was very different from the world people know today. The atmosphere contained very little free oxygen. Instead, it was made mostly of gases that came from volcanic eruptions and from chemical reactions taking place inside the young planet. Carbon dioxide, methane, water vapor, nitrogen, and several other gases were much more common than oxygen. If a modern human had somehow traveled back to that time, breathing the air would not have been possible. The atmosphere could not support human life because oxygen was almost absent. Scientists believe that the earliest forms of life appeared in the oceans. Water offered protection from harmful radiation coming from the sun, because the young Earth did not yet have an ozone layer. Inside these ancient seas, simple living cells slowly developed. Over many millions of years, life became more varied. Some microbes learned to obtain energy from chemicals found in rocks or dissolved in seawater. Others eventually developed the remarkable ability to capture energy from sunlight. Photosynthesis became one of the most important biological processes in Earth's history. During photosynthesis, certain microbes absorbed sunlight and used its energy to combine water and carbon dioxide into food. As a result of this process, oxygen was released into the surrounding water and later into the atmosphere. Today, plants perform this same basic process, but plants did not yet exist when photosynthetic microbes first appeared. The earliest known organisms to carry out this type of photosynthesis were cyanobacteria, sometimes called blue-green algae, even though they are actually bacteria. Cyanobacteria were simple single-celled organisms. They did not have roots, stems, leaves, or flowers. Even without these structures, they were able to survive in many different environments. They spread across shallow seas where sunlight was available. They often formed large communities that covered rocks and other underwater surfaces. Layer after layer of these microbial communities slowly trapped tiny particles of sediment. As the layers built up over very long periods, they formed rocky structures known as stromatolites. Some stromatolites are among the oldest direct signs of life found on Earth. The fossil record shows that stromatolites were much more common in the distant past than they are today. Ancient stromatolites provide important evidence that photosynthetic microbes were already active billions of years ago. Scientists study these fossils to learn about early life and the conditions that existed in ancient oceans. The layered appearance of stromatolites reveals repeated growth by microbial communities over long stretches of time. When cyanobacteria released oxygen through photosynthesis, the oxygen did not immediately collect in the atmosphere. Instead, it reacted with substances already present in the oceans. Large amounts of dissolved iron floated in seawater during this period. Oxygen combined with the iron to form iron oxides. These compounds eventually settled onto the ocean floor, creating thick deposits that later became important sources of iron ore. Many of the iron formations mined today began forming during this ancient period when oxygen first entered Earth's oceans. For a long time, nearly all the oxygen produced by photosynthetic microbes was used up in these chemical reactions. As more oxygen continued to be produced, increasing amounts of iron became locked away in rocks. Gradually, fewer dissolved minerals remained available to react with oxygen. This allowed oxygen to begin collecting first in seawater and later in the atmosphere. This slow increase in atmospheric oxygen marked one of the greatest environmental changes in Earth's history. Scientists often refer to this event as the Great Oxygenation Event. It happened over an extremely long period rather than during a single moment. Oxygen levels rose gradually as countless generations of photosynthetic microbes continued carrying out photosynthesis every day. No single group of microbes changed the atmosphere by itself. The combined work of uncountable numbers of microorganisms over many millions of years transformed the air surrounding the planet. The rise of oxygen brought major changes for existing life. Many early organisms had evolved in environments without oxygen. For some of them, oxygen was harmful because their cells were not adapted to handle it. As oxygen levels increased, many of these organisms disappeared from places where oxygen became common. Others survived by living in environments where oxygen remained very low, such as deep underground or within certain layers of mud and sediment. Even today, some microorganisms live successfully in places where oxygen is absent. At the same time, the growing presence of oxygen created opportunities for new kinds of life. Some organisms evolved ways to use oxygen to release energy from food much more efficiently. This process, known as aerobic respiration, allowed cells to produce larger amounts of usable energy. Having more energy available supported the development of larger and more complex living organisms over time. Although this transformation did not happen quickly, the availability of oxygen opened many new possibilities for biological evolution. Another important result of rising oxygen levels involved the formation of the ozone layer. Some oxygen molecules high in the atmosphere were changed by sunlight into ozone. Ozone formed a protective layer that absorbed much of the harmful ultraviolet radiation coming from the sun. Before this protective shield developed, strong ultraviolet radiation made conditions on the land much more difficult for living organisms. As the ozone layer became stronger, it reduced the amount of harmful radiation reaching Earth's surface. This greater protection eventually made it easier for life to expand into environments outside the oceans. Water had served as a natural shield against harmful radiation for early organisms. As atmospheric conditions changed and the ozone layer became established, more living things could survive closer to the surface and eventually on land. This opened another important chapter in the history of life. The process through which photosynthetic microbes changed the atmosphere required remarkable patience on a planetary scale. These microorganisms lived, reproduced, and died continuously for countless generations. Each individual cell contributed only a tiny amount of oxygen. Yet the combined work of endless numbers of microbes over vast stretches of time slowly altered the chemistry of the oceans and the atmosphere. This gradual change demonstrates how small biological activities, repeated over very long periods, can reshape an entire planet. The success of cyanobacteria depended on several environmental conditions. They needed sunlight because photosynthesis requires light energy. They also needed water and carbon dioxide. Ancient shallow seas provided all these requirements. Sunlight could easily penetrate the upper layers of water, allowing microbial communities to grow in large numbers. Coastal regions, tidal flats, and warm marine environments became especially favorable places for these organisms. Not every photosynthetic microbe lived in exactly the same way. Different species adapted to different temperatures, salt levels, and light conditions. Some formed floating communities near the water's surface, while others attached themselves to rocks beneath shallow water. These differences helped photosynthetic microbes spread across many parts of the ancient oceans. The early atmosphere probably experienced many gradual changes, while oxygen accumulated. Volcanic eruptions continued releasing gases into the air. Chemical reactions involving rocks, oceans, and the atmosphere also continued. Photosynthetic microbes became one of the strongest biological influences on these natural processes because they constantly removed carbon dioxide during photosynthesis while releasing oxygen. Carbon dioxide played an important role in Earth's climate because it trapped heat within the atmosphere. As photosynthetic microbes absorbed carbon dioxide, they affected the balance of atmospheric gases. Some of the carbon became stored inside living cells. After microbes died, part of this carbon settled into sediments on the ocean floor. Over immense periods, some of these carbon-rich materials became buried beneath newer layers of sediment, locking away carbon that had once existed in the atmosphere. Scientists continue studying ancient rocks to understand exactly how oxygen levels changed through time. Rocks preserve chemical clues that reveal conditions existing when they formed. Certain minerals can only develop in the presence of oxygen, while others form only when oxygen is absent. By examining these minerals, researchers reconstruct the history of Earth's atmosphere and estimate when oxygen became increasingly common. Modern technology allows scientists to analyze tiny amounts of chemical material trapped inside ancient rocks. These studies provide evidence supporting the idea that oxygen accumulated gradually, rather than appearing suddenly. Different regions of the planet may also have experienced rising oxygen levels at different times depending on local environmental conditions. Photosynthetic microbes also influence nutrient cycles within the oceans. As they grew and multiplied, they absorbed nutrients from seawater. When they died, their remains sank toward the ocean floor, carrying nutrients with them. Other microorganisms broke down this organic material, releasing nutrients that could be used again by new generations of life. These repeating cycles became an important part of Earth's biological systems. The activity of cyanobacteria affected marine ecosystems in many ways. Their ability to produce food through photosynthesis supported other organisms that could not make their own food. Tiny organisms fed directly on microbial cells, while larger organisms eventually fed on those smaller consumers. Over long periods, increasingly complex food webs developed within the oceans. Even today, cyanobacteria remain important members of Earth's ecosystems. They live in oceans, lakes, rivers, wetlands, deserts, hot springs, polar environments, and even inside rocks. Some species can survive in conditions that would be difficult for many other forms of life. Their long evolutionary history has allowed them to develop many different ways of adapting to changing environments. Scientists studying modern cyanobacteria gain valuable insights into ancient life. Although present-day species have continued evolving, they still perform photosynthesis, using the same basic chemical process that helped reshape Earth's atmosphere long ago. Observing these living organisms helps researchers understand how early microbial communities may have behaved in ancient oceans. The oxygen produced by photosynthetic microbes eventually became essential for animals, plants, fungi, and many microorganisms. Complex multicellular life depends on oxygen to meet its energy needs. Without the earlier work of photosynthetic microbes, such forms of life would likely never have developed in the way they did. The atmosphere had to change before many later evolutionary developments became possible. The fossil evidence preserved in stromatolites remains one of the strongest links to this distant chapter of Earth's history. Some living stromatolites still exist today in a few special environments where conditions allow microbial communities to grow without being heavily disturbed. These living examples provide scientists with opportunities to observe processes similar to those that occurred in ancient seas. Researchers also investigate microscopic fossils, chemical signatures, and ancient sediments to better understand the spread of photosynthetic microbes. Every new discovery helps improve knowledge about how Earth's atmosphere developed through natural biological activity. The evidence collected from rocks around the world continues to strengthen the understanding that tiny microorganisms played a central role in transforming the planet over immense spans of time. The history of photosynthetic microbes shows that Earth's atmosphere has never remained completely unchanged. The mixture of gases surrounding the planet has shifted many times because of interactions among living organisms, volcanoes, oceans, rocks, sunlight, and countless chemical reactions. Photosynthetic microbes became one of the most powerful living forces influencing this long history because they continuously released oxygen while removing carbon dioxide from their surroundings. As oxygen became more abundant, Earth's environment continued changing in many connected ways. New ecosystems formed, different groups of organisms evolved, and biological diversity gradually increased. The atmosphere, oceans, and land remained closely connected through ongoing physical, chemical, and biological processes that continued shaping the planet across vast stretches of geological time. Cyanobacteria are among the oldest known living organisms on Earth. These tiny living cells have existed for an extremely long time and played an important part in changing the history of the planet. Even though they are very small and can only be seen clearly with a microscope, their activities slowly changed the air, the oceans and the surface of Earth. Before cyanobacteria became common, Earth was a very different place. The atmosphere contained very little oxygen, and the kinds of living things that could survive were limited. The slow work of cyanobacteria over an enormous length of time added oxygen to the atmosphere and changed the conditions of the planet. The early Earth formed about 4,500 million years ago. During its earliest history, the surface was very hot. Volcanoes were active in many places, and molten rock covered large areas. As the planet slowly cooled, a solid crust formed. Water vapor in the atmosphere cooled and fell as rain over long periods, creating the first oceans. These early oceans became places where the first simple forms of life appeared. Scientists continue to study exactly how life began, but evidence suggests that tiny single-celled organisms were already living in the oceans more than 3,500 million years ago. Among these ancient living organisms were cyanobacteria. They were simple cells without a nucleus, making them part of a group known as bacteria. Even though they were simple in structure, they possessed an important ability that would eventually affect the entire planet. They could use sunlight as a source of energy. This process is called photosynthesis. During photosynthesis, cyanobacteria used sunlight, water, and carbon dioxide to produce food for themselves. At the same time, they released oxygen as a waste product. Today, oxygen is a normal part of the atmosphere, and many living things depend on it every moment. Animals, plants, fungi, and many tiny organisms need oxygen to survive. Early Earth was different. The atmosphere contained gases such as carbon dioxide, nitrogen, methane, water vapor, and other volcanic gases. Free oxygen was almost completely absent. Because oxygen was so rare, the earliest living organisms evolved in an environment that did not depend on oxygen. Many of them gained energy through chemical reactions that worked without it. When cyanobacteria first appeared, the oxygen they produced did not immediately fill the atmosphere. Instead, much of it entered the oceans. The seawater contained large amounts of dissolved iron. Oxygen reacted with this iron, creating iron oxides that settled onto the ocean floor. Over many millions of years, these deposits built thick layers of rock. Today, these rocks are known as banded iron formations. They provide important evidence that oxygen was being produced long before it became common in the atmosphere. The formation of these iron-rich rocks continued for a very long time. As cyanobacteria kept producing oxygen every day, more dissolved iron reacted with it. Eventually, much of the available iron in the oceans became locked inside minerals. Once less dissolved iron remained, increasing amounts of oxygen could stay in the water instead of being used in chemical reactions. Gradually, oxygen began to move from the oceans into the atmosphere. This long period of increasing atmospheric oxygen is known as the Great Oxygenation Event. Scientists believe that it began about 2,400 million years ago. It did not happen suddenly over a few years or even a few thousand years. It was an extremely slow process that lasted for many millions of years. During this time, oxygen levels slowly rose, although they were still much lower than they are today. The increase in oxygen changed Earth's environment in many ways. Many chemical reactions that had been common for billions of years began to change. Minerals on land reacted with oxygen and formed new compounds. Rocks exposed to the air developed different colors because of oxidation. These changes can still be seen in ancient rocks studied by geologists today. Not every living organism benefited from the rising oxygen. For many early forms of life, oxygen was harmful. Their cells had evolved in an environment where oxygen was almost absent. When oxygen levels increased, it damaged many of their biological processes. Some organisms disappeared completely, while others survived only in places where oxygen remained scarce, such as deep underground, inside sediments, or in environments where oxygen could not easily reach. Some bacteria living today still avoid oxygen because it is harmful to them. These organisms provide scientists with clues about what some of Earth's earliest life may have been like before oxygen became common. They continue to live in environments that resemble some of the conditions found on ancient Earth. While oxygen was dangerous for certain organisms, other forms of life gradually evolved ways to use it. Oxygen allowed cells to release much more energy from food than earlier methods could provide. This greater supply of energy supported the development of more active and more complex living organisms. Over many millions of years, this increased energy availability helped life become more diverse. Cyanobacteria themselves continued to spread into many different environments. They lived in shallow coastal waters, lakes, rivers, and warm pools. Some floated freely in the water, while others formed large communities attached to rocks or sediments. Many lived in places where sunlight was strong because sunlight was necessary for photosynthesis. One of the most important structures created by cyanobacteria is called a stromatolite. Stromatolites formed when layers of cyanobacteria trapped tiny particles of sand, mud, and other materials. As new layers of bacteria grew on top of older ones, these structures slowly became larger. Over very long periods, they formed rounded or layered rocky shapes. Some ancient stromatolites are more than 3 billion years old. They are among the oldest clear signs of life found on Earth. Modern stromatolites still exist in a few places around the world. Scientists study them because they provide valuable information about how ancient cyanobacteria lived. Although modern examples grow under different conditions than many ancient ones, they help researchers understand how these communities formed and survived. The success of cyanobacteria came partly from their ability to use sunlight directly. Every day, sunlight provided energy for photosynthesis. As long as water, carbon dioxide, and sunlight were available, these organisms could continue growing and producing oxygen. Since sunlight reached enormous areas of the oceans, cyanobacteria had many opportunities to spread across large regions. The carbon dioxide used during photosynthesis also affected Earth's climate over long periods. As cyanobacteria removed carbon dioxide from the atmosphere and oceans, some of the carbon became stored inside living cells or buried in sediments after the organisms died. This slowly changed the balance of gases in the atmosphere. Scientists believe these changes influenced Earth's climate in different ways during ancient times. Life during the time of cyanobacteria remained simple for a very long period. Most organisms consisted of only one cell. They reproduced by dividing into 2 new cells. Under favorable conditions, their populations could increase quickly. Even though each individual organism was extremely small, the combined activity of countless numbers of cyanobacteria across the oceans produced enormous amounts of oxygen over geological time. The early oceans contained many different chemicals that reacted with oxygen. Besides iron, sulfur, and other elements also combined with oxygen through natural chemical reactions. These reactions delayed the buildup of oxygen in the atmosphere. Cyanobacteria had to continue producing oxygen for many millions of years before enough remained free to accumulate in the air. Scientists study ancient rocks from many parts of the world to understand the Great Oxygenation Event. They examine the minerals inside these rocks, measure the chemical elements they contain, and analyze tiny differences in their composition. These studies help researchers estimate how much oxygen existed at different times in Earth's history. Although many details are still being investigated, the evidence strongly supports the idea that cyanobacteria were the main source of Earth's early oxygen. Fossils of cyanobacteria are usually very small because the organisms themselves are microscopic. Many fossil remains appear as tiny layers, filaments, or rounded shapes preserved inside ancient rocks. Modern technology allows scientists to study these fossils in great detail. Powerful microscopes reveal structures that help identify ancient bacterial communities and provide information about their growth patterns. Cyanobacteria also developed ways to survive changing environmental conditions. Some species formed protective outer coverings that reduced damage from drying or harmful conditions. Others produced special cells that allowed them to survive periods when water became scarce. These abilities helped them remain successful in many environments across the ancient world. Their growth depended on the availability of nutrients as well as sunlight. Elements such as phosphorus, nitrogen, and iron supported important biological processes inside their cells. Rivers, volcanic activity, and weathering of rocks supplied many of these nutrients to lakes and oceans. When nutrients were abundant, cyanobacterial populations often increased. The relationship between cyanobacteria and the changing atmosphere continued over countless generations. Every new generation carried out photosynthesis, releasing small amounts of oxygen. Although each individual cell contributed only a tiny amount, the combined activity of immense populations over millions of years gradually transformed Earth's atmosphere into one that contained increasing levels of oxygen. The earliest plant-like organisms were made of only a single cell. Each cell was a complete living organism that carried out every activity needed for survival. It gathered nutrients, responded to changes in its surroundings, reproduced, repaired itself, and produced energy. These single-celled organisms were extremely small and could only be seen with powerful microscopes. Even though they were tiny, they became some of the most successful living things on Earth, because they multiplied quickly and lived in many different aquatic environments. The ancient oceans of Earth were very different from the oceans of today. The water contained dissolved minerals that came from volcanic activity and from rocks that were slowly broken down by rain and flowing water. The atmosphere had very little oxygen. Instead, gases such as carbon dioxide, methane, water vapor, and nitrogen were much more common. The sun was slightly dimmer than it is today, but enough sunlight reached the surface of the oceans to support photosynthesis in shallow water. These conditions created places where early photosynthetic organisms could survive and slowly spread. Many scientists believe that cyanobacteria were among the earliest organisms to perform oxygen-producing photosynthesis. Cyanobacteria are bacteria, not plants, but they share an important ability with plants because they use sunlight to make food. They lived in shallow seas, lakes, coastal waters, and other wet places where sunlight could reach them. As they grew, they often formed large colonies that spread across rocks and soft sediments. Some colonies trapped tiny particles carried by water. Layer after layer built up over very long periods, forming structures known as stromatolites. Some stromatolites created by ancient cyanobacteria still exist today and provide valuable evidence about the earliest history of life. Each cyanobacterial cell contained special molecules that absorbed light energy from the sun. This energy powered a series of chemical reactions inside the cell. Water molecules were broken apart, hydrogen was used to build sugars, and oxygen was released as a waste product. At first, this oxygen reacted with iron and other materials dissolved in the oceans. Large amounts of iron combined with oxygen and settled onto the ocean floor, creating thick deposits that can still be found in many parts of the world. Only after these materials had reacted with oxygen for a very long time did oxygen begin to collect in the atmosphere. The slow increase of oxygen changed Earth's environment in many ways. Organisms that could survive in oxygen-rich conditions became more common. New forms of life slowly evolved. The ozone layer gradually formed high in the atmosphere as oxygen molecules interacted with sunlight. This ozone layer blocked much of the harmful ultraviolet radiation coming from the sun. As a result, living things had greater protection from damaging radiation, making it easier for life to survive in shallow water and eventually on land. Although cyanobacteria were extremely important, they were still bacteria. They did not have a nucleus inside their cells. Their genetic material floated freely within the cell. They also lacked many of the complex internal structures that later appeared in more advanced organisms. Over a very long period, another important step took place in the history of life. Cells became more complex and developed a nucleus surrounded by a membrane. These organisms are called eukaryotes. The appearance of eukaryotic cells opened the way for greater biological complexity. Scientists think that one of the most important events in evolution happened when a large single-celled organism surrounded a photosynthetic cyanobacterium without digesting it. Instead of being destroyed, the smaller organism remained alive inside the larger cell. Both organisms benefited from this arrangement. The larger cell provided protection and access to nutrients. The smaller photosynthetic cell continued producing food using sunlight. As generation after generation passed, the relationship became permanent. The photosynthetic cell gradually lost its independence and became an organelle known as the chloroplast. Chloroplasts are found in the cells of plants and algae today. They contain chlorophyll, the green pigment that captures sunlight for photosynthesis. The presence of chloroplasts allows plants and algae to make their own food instead of depending on other organisms for energy. Modern studies of chloroplast DNA show strong evidence that chloroplasts evolved from ancient cyanobacteria. This discovery has helped scientists understand the close relationship between bacteria and the earliest plant-like organisms. The earliest eukaryotic organisms with chloroplasts were still very simple. Most consisted of only one cell. They floated freely in oceans, lakes, rivers, and ponds. Ocean currents carried them from one place to another. Some lived close to the surface where sunlight was strongest, while others remained at slightly deeper levels where enough light still reached them. Their ability to carry out photosynthesis allowed them to grow without eating other living things. They only needed sunlight, water, carbon dioxide, and dissolved minerals. These simple photosynthetic eukaryotes reproduced mainly by dividing into two identical cells. Before division, each cell copied its genetic material. The cell then split into two daughter cells, each receiving a complete set of genetic instructions. This process allowed populations to increase rapidly when conditions were favorable. Warm temperatures, abundant sunlight, and enough nutrients encouraged fast growth. During less favorable conditions, growth slowed until the environment improved. Random genetic changes occasionally appeared as cells reproduced. Some of these changes helped organisms survive better in their surroundings. Cells with useful traits produced more offspring, allowing those traits to become more common over many generations. This gradual process of evolution slowly produced greater diversity among early photosynthetic organisms. Different groups adapted to different temperatures, light levels, water chemistry and nutrient availability. Some early photosynthetic organisms remained solitary throughout their lives. Others began living together in loose colonies. Colonial living provided several advantages. Neighbouring cells could remain close to one another while still carrying out their own individual activities. Colonies sometimes became large enough to be visible without magnification. Although each cell still functioned independently, living together created opportunities for greater cooperation among neighbouring cells. Over time, certain colonies became more organised. Some cells specialised in particular jobs, while others carried out different tasks. This gradual specialisation eventually led toward multicellular organisms. Instead of every cell doing exactly the same work, different cells began contributing to the survival of the entire organism in different ways. This represented an important step toward the evolution of larger and more complex forms of life. The earliest simple algae probably evolved from these ancient photosynthetic eukaryotes. Algae include many different kinds of organisms that perform photosynthesis but are not considered true land plants. Early algae lived entirely in water. Some remained single-celled while others became multicellular. Their bodies lacked true roots, stems, leaves, flowers, seeds, and vascular tissues. Instead, they absorbed water and dissolved nutrients directly across their surfaces. The bodies of simple algae are called thalli. A thallus is a body that does not contain the specialized organs found in land plants. Even though this body structure appears simple, it functions effectively in aquatic environments where water surrounds every part of the organism. Nutrients and gases move directly into cells from the surrounding water, reducing the need for specialized transport systems. The earliest algae probably appeared in oceans before spreading into freshwater environments. Rivers, lakes, ponds, marshes, and shallow pools offered many suitable habitats. Different environments presented different challenges. Salt levels, nutrient concentrations, water movement, temperature, and light availability varied from place to place. As algae adapted to these different conditions, many separate groups evolved with their own unique characteristics. Green algae eventually became especially important because scientists believed they share a close evolutionary relationship with land plants. Their cells contain chlorophyll, similar to that found in modern plants. They store food in the form of starch and possess many other biological features that later appeared in land plants. These similarities provide strong evidence that the ancestors of modern plants evolved from ancient green algae that lived in freshwater habitats. The earliest green algae displayed many different shapes. Some consisted of single round cells drifting freely through the water. Others formed long chains of connected cells. Some produced flat sheets only a few cells thick. Others developed branching filaments attached to rocks or other surfaces. These different body forms allowed algae to occupy many habitats while receiving enough sunlight for photosynthesis. Many algae attached themselves to rocks using simple structures that anchored them in place. These attachments prevented them from being washed away by waves or currents. Unlike the roots of modern plants, these structures mainly served as anchors rather than absorbing water and nutrients. Since algae lived underwater, every part of their bodies remained surrounded by water, allowing nutrients to enter directly through their cell surfaces. Sunlight played a central role in the lives of these early plant-like organisms. Photosynthesis depended on light energy, so algae and other photosynthetic organisms often grew where sunlight could easily penetrate the water. Clear, shallow water provided especially favorable conditions. In deeper water, light became weaker. Different pigments inside algal cells helped absorb available light, allowing some species to live at greater depths than others. The oceans contained dissolved carbon dioxide that algae used during photosynthesis. They also absorbed minerals such as nitrogen, phosphorus, sulfur, magnesium, calcium, and potassium. These nutrients supported growth, cell division, and the production of proteins, genetic material, and other important substances. When nutrients became scarce, growth slowed until fresh supplies entered the water through rivers, ocean currents, volcanic activity, or the breakdown of organic matter. Early algae formed the foundation of ancient aquatic food webs. Although they were very small, countless numbers of them lived in the oceans. Tiny animals fed on these algae. Larger animals fed on the smaller animals, and increasingly complex food chains gradually developed. In this way, photosynthetic organisms converted sunlight into food that supported many other forms of life. The growth of algae also influenced the movement of carbon through Earth's environment. During photosynthesis, algae removed carbon dioxide from the water and atmosphere. Some of this carbon became part of their bodies. When algae died, part of their remains settled to the bottoms of lakes and oceans. Some of this organic material decomposed, while some became buried beneath layers of sediment. Over extremely long periods, this burial helped store carbon within Earth's crust. The remains of ancient algae have provided scientists with valuable evidence about the history of life. Tiny fossils preserved in ancient rocks reveal the shapes and structures of early photosynthetic organisms. Chemical evidence preserved within rocks also provides clues about ancient biological activity. Scientists carefully study these fossils using microscopes and advanced laboratory techniques to understand when different groups appeared and how they evolved over time. Some cells began producing substances that held neighboring cells together more firmly. These natural materials strengthened the bodies of multicellular algae and reduced the chance that waves or currents would pull them apart. Stronger connections between cells also allowed organisms to grow into longer filaments, wider sheets, and more complex branching forms. Although these bodies were still simple, they represented another important stage in biological development. Growth in multicellular algae usually occurred when cells near the ends of the body divided repeatedly. New cells formed one after another, increasing the length or width of the organism. In some species, growth took place across much of the body instead of only at the tips. Green algae are among the most important living organisms for understanding the history of plants. Scientists have spent many years studying them because they provide valuable information about how the first plants that lived on land may have developed. Green algae are still alive today in many different environments, and they continue to grow in lakes, rivers, ponds, wetlands, oceans, and even on damp rocks, tree bark, and moist soil. Although they are simple in many ways, they carry many important features that connect them with all the plants that now cover forests, grasslands, mountains, deserts, and gardens across the world. The story of green algae reaches back into the distant past of Earth. Long before forests covered the continents, long before flowers appeared, and long before large trees spread across the land, green algae were already growing in water. They were part of the slow changes that helped shape life on the planet. Scientists believe that one group of green algae eventually gave rise to the earliest land plants. This idea is supported by many different kinds of evidence collected from fossils, living species, cell structure, genetics, reproduction, and development. Green algae belong to a larger group of living things that use sunlight to make their own food through photosynthesis. Like all green plants, they contain chlorophyll. Chlorophyll is the green pigment that captures sunlight and uses its energy to produce sugars from water and carbon dioxide. These sugars provide food for the organism and store energy that can later be used for growth and reproduction. The green colour of these algae comes mainly from chlorophyll A and chlorophyll B. These are the same major pigments found in land plants today. This shared feature is one of the strongest pieces of evidence showing that green algae and land plants are closely related. Many other kinds of algae contain different pigments that give them brown, red, golden or blue-green colours. But green algae share the same main pigments as modern plants. The cells of green algae also contain chloroplasts. Chloroplasts are the small structures inside cells where photosynthesis takes place. Inside these chloroplasts, sunlight is turned into chemical energy. Scientists have found that the chloroplasts of green algae and those of land plants are very similar in both their structure and their genetic material. This similarity suggests that they inherited these chloroplasts from a common ancestor that lived in the distant past. Green algae store food in the form of starch. This is another important feature they share with land plants. Starch acts as stored energy that can be used when sunlight is limited or when the organism needs extra energy for growth and reproduction. Many other groups of algae store different kinds of food materials instead of starch. The fact that green algae and land plants both store starch in similar ways provides another clue that they belong to the same branch of life's history. The walls surrounding the cells of green algae are usually made mainly from cellulose. Cellulose is a strong material that gives support and shape to the cell. Modern land plants also use cellulose as the main building material for their cell walls. Cellulose allows cells to remain firm while still allowing water and nutrients to move where they are needed. This shared feature is another important connection between green algae and land plants. Green algae come in many different shapes and sizes. Some consist of only a single cell that cannot be seen without a microscope. Others form colonies made of many cells working together. Some grow into long threads called filaments. Others develop flat sheets or more complex branching forms. This wide variety shows that green algae experimented with many ways of growing long before plants moved onto land. Single-celled green algae carry out all the activities needed for life within one tiny cell. They absorb sunlight, make food, grow, reproduce, and respond to changes in their surroundings. These tiny organisms may appear simple, but each cell contains many carefully organized structures that allow it to survive. Colonial green algae are made of many cells living together. In some colonies, every cell performs the same tasks. In other colonies, some cells begin to take on slightly different roles. This simple sharing of work may have been one step toward the more complex bodies found later in land plants. Filamentous green algae grow as long chains of connected cells. Each cell remains attached to the next, forming thread-like bodies that may float in water or attach themselves to rocks, plants, or other surfaces. These filaments allow the algae to spread across larger areas while remaining connected. Scientists believe that this type of growth helped prepare some algae for the challenges of living outside water. Some green algae have bodies that branch into many directions. These branching forms increase the area exposed to sunlight, allowing more photosynthesis to take place. They also provide more places where reproduction can occur. Branching became an important feature later seen in the stems and leaves of land plants. Green algae are found in almost every part of the world where enough moisture is available. Many species live in freshwater environments such as ponds, lakes, streams, rivers, marshes and wetlands. Others grow in saltwater along ocean shores or in shallow coastal waters. Some survive in very cold regions, while others grow in warm tropical climates. Many green algae grow attached to rocks beneath flowing water. Others live on submerged logs, aquatic plants, shells or muddy bottoms. Some float freely in open water where sunlight reaches them. Tiny species may drift with water currents as part of the plankton community, providing food for many small animals. Green algae can also live outside water if enough moisture is present. Some grow on damp tree trunks, shaded rocks, walls, roofs, caves, or moist soil after rainfall. Others live together with fungi to form lichens. In these partnerships, the algae produce food through photosynthesis, while the fungus provides protection and helps retain moisture. One reason green algae have survived for such a long time is their ability to adapt to many different environments. Different species have developed different ways of handling changes in light, temperature, water, and nutrients. Some survive periods of drying by forming tough protective coverings. Others produce special resting cells that remain inactive until better conditions return. The life cycles of green algae vary greatly from one species to another. Some reproduce by simple cell division. During this process, one cell grows larger and then splits into two new cells. Each new cell continues growing independently. This method allows populations to increase quickly when conditions are favorable. Many green algae also reproduce by producing spores. Spores are special cells that can grow into new individuals without joining with another cell. Spores are often released into water where they settle in new places and begin growing if conditions are suitable. Sexual reproduction also occurs in many green algae. During this process, two reproductive cells join together to form a new individual. Sexual reproduction creates new combinations of genetic material. These new combinations increase variation within populations, giving species a better chance of surviving changing environments. Different species of green algae show different stages in the development of sexual reproduction. In some species, the joining cells are almost identical. In others, one reproductive cell is larger while the other is smaller. These gradual changes help scientists understand how more specialized forms of reproduction developed over time. Scientists divide green algae into several major groups based on their characteristics and their genetic relationships. Among these groups, one branch known as the charophytes has received special attention because it appears to be the closest living relative of land plants. Studies of their genes, cells, and life cycles show many similarities with the earliest plants that eventually spread across the continents. Charophytes include several living groups that still grow mainly in freshwater habitats. Although they remain aquatic organisms, they possess many features once believed to exist only in land plants. These shared features help scientists understand how the first plants gradually became capable of surviving outside water. One important shared feature involves the way cells divide. During cell division, both charophytes and land plants use special structures that guide the formation of new cell walls. This process is highly organized and allows tissues to grow in an orderly manner. Other groups of algae divide their cells differently. Scientists have also discovered that the enzymes involved in making cellulose are very similar in charophytes and land plants. These enzymes help build strong cell walls that support the organism while allowing continued growth. Communication between neighboring cells is another shared feature. Tiny channels connect adjacent cells in both charophytes and land plants. These channels allow materials and signals to pass directly from one cell to another. This coordination helps groups of cells function together more effectively. Green algae also possess many genes that resemble those found in land plants. Modern genetic studies have shown that numerous important genes involved in growth, development, stress response, and reproduction were already present before plants moved onto land. Land plants later adapted these existing genes to meet the new challenges of terrestrial life. Scientists compare DNA from many living organisms to understand evolutionary relationships. These genetic studies repeatedly show that land plants share their closest relationship with charophyte green algae rather than with any other group of algae. This conclusion is supported by large amounts of genetic evidence collected from many species. Living on land required many new abilities. Organisms had to avoid drying out, support themselves without floating in water, protect reproductive cells, obtain water from the ground, and survive changing weather. Many of the features needed for these challenges already existed in simple forms among the ancestors found within green algae. Some green algae occasionally experience drying when ponds become shallow or when water levels fall. These conditions may have helped natural selection favor individuals that could survive temporary water loss. Over long periods of time, these survival abilities became increasingly useful for organisms beginning to live on moist land. Exposure to sunlight outside water also brought new challenges. Without water to filter sunlight, harmful ultraviolet radiation became stronger. Some green algae developed protective chemicals that reduced damage from this radiation. Similar protective systems are found in land plants today. Changes in temperature also became more important outside water. Water usually changes temperature more slowly than air. Organisms beginning to spend time on land had to cope with greater daily temperature changes. Some green algae already possessed ways to respond to these changing conditions. Nutrients are obtained differently on land than in water. Aquatic organisms absorb many dissolved nutrients directly from surrounding water. On land, nutrients must often be taken from soil. The earliest land plants gradually developed structures that improved nutrient absorption, while still retaining many features inherited from their algal ancestors. Water movement also became more difficult outside aquatic environments. In water, cells remain surrounded by moisture. On land, water can quickly evaporate. This created pressure for the development of structures that reduced water loss while allowing continued growth and photosynthesis. The earliest land plants probably remained close to wet environments where moisture was always available. They may have grown along riverbanks, lake shores, marshes, wetlands, or damp soil. Living in these places allowed gradual adjustment to life outside permanent water. Green algae played an important role in Earth's ecosystems long before land plants appeared. Through photosynthesis, they produced oxygen and removed carbon dioxide from the atmosphere. They also formed the foundation of many aquatic food chains by providing food for tiny animals, which were then eaten by larger organisms. Even today, green algae remain essential members of freshwater and marine ecosystems. Small animals feed directly on them. Fish feed on those animals. Larger predators feed on the fish. In this way, energy captured from sunlight moves through many levels of life. Green algae also help cycle nutrients through aquatic ecosystems. As they grow, they absorb minerals from water. When they die, decomposers break down their bodies, releasing nutrients that become available for other living organisms. Some species of green algae live inside other organisms in helpful relationships. Certain tiny animals contain green algae within their bodies. The algae provide food through photosynthesis, while the animals offer protection and access to nutrients. Many scientists study living green algae because fossils from very ancient times are often incomplete. Soft-bodied organisms usually decay quickly after death, leaving few traces behind. By examining living species, researchers can better understand the possible appearance and biology of ancient ancestors. The fossil record nevertheless provides important evidence that green algae have existed for an extremely long time. Fossil remains and chemical evidence found in ancient rocks suggest that green algae were already widespread in Earth's waters hundreds of millions of years before forests developed across the continents. Modern technology has greatly increased scientific knowledge about green algae. Powerful microscopes reveal tiny details inside cells that earlier scientists could not observe. DNA sequencing allows researchers to compare genes from different organisms with remarkable accuracy. These methods continue to strengthen the evidence linking green algae with land plants. Laboratory experiments also help scientists understand how green algae respond to drying, changes in light, nutrient shortages, and temperature changes. These studies reveal that many protective systems found in land plants already existed in simpler forms among their algal relatives. Some green algae produce special protective substances that help their cells remain stable during periods of stress. These substances reduce damage caused by drying, intense sunlight, or changing temperatures. Similar protective chemicals are also found in land plants. Researchers have discovered that certain proteins involved in detecting environmental changes are shared between green algae and land plants. These proteins help cells sense light, gravity, temperature, and other conditions so that appropriate responses can occur. The movement of reproductive cells also varies among green algae. Some species release swimming reproductive cells equipped with tiny whip-like structures called flagella. These allow the cells to move through water until they reach another reproductive cell. Other species have lost these swimming cells and instead depend on different methods of reproduction. This gradual change helps scientists understand how reproduction adapted as plants moved onto land where free water became less available. Green algae also display many different growth patterns during their life cycles. Some spend most of their lives as single cells, while others remain multicellular throughout their existence. This diversity provides valuable examples of how increasing complexity may have developed over long periods of evolutionary history. Many school textbooks describe green algae as simple organisms, but their biology is surprisingly rich. Each species has its own life cycle, environmental needs, methods of reproduction, and cellular organization. Thousands of living species have been identified, and scientists continue discovering new ones in different habitats around the world. Human beings also benefit from green algae in many ways. Scientists use certain species in biological research because they grow quickly and are easy to study. They help researchers understand photosynthesis, genetics, cell division, and many other biological processes that apply to plants more broadly. Some green algae are used in wastewater treatment because they absorb nutrients that would otherwise pollute rivers and lakes. As they grow, they remove nitrogen and phosphorus from the water, helping improve water quality. The evolution of roots, stems, and primitive leaves was one of the most important stages in the early history of plants. These structures allowed plants to survive in places that were very different from the shallow water where their ancestors first lived. Early plant life began in water, where every part of the plant was surrounded by moisture. Water supported the body of the plant, carried minerals, and made it easy for cells to exchange gases. When some plant ancestors slowly began living on land, they entered an environment that brought many new challenges. The ground was dry in many places, sunlight was stronger, the wind could remove water from their bodies, and gravity pulled on every part of the plant without the support of water. To remain alive, these early plants gradually developed new body parts that helped them meet these challenges. The earliest land plants were very simple. They did not have true roots, true stems, or true leaves. Their bodies were made of small green parts that carried out photosynthesis while staying close to the damp ground. These plants depended on places where water was always available because they could not collect water from deep inside the soil. They also had no strong support to help them grow very tall. Their small size helped them survive because every part of the plant remained close to moisture. As many generations passed, small changes appeared in different groups of plants. Some of these changes helped certain plants survive better than others. Plants with useful features produced more offspring, and these helpful features became more common over long periods of time. This slow process led to the appearance of the first structures that later became roots, stems, and leaves. Before true roots existed, many early land plants used thin hair-like structures called rhizoids. Rhizoids were much simpler than roots. Their main job was to hold the plant in place so that rain, flowing water, or wind could not easily remove it from the ground. Rhizoids also helped absorb small amounts of water from the soil, but they could not move large amounts of water through the plant. They had no special tissues for carrying water or food over long distances. Because of this, plants that depended only on rhizoids remained small and lived in wet places. The appearance of true roots changed the lives of plants in many ways. Roots were able to grow downward into the soil. They held plants firmly in the ground and allowed them to survive strong winds much better than before. As roots spread through the soil, they reached water that was hidden below the surface. They also absorbed important minerals that plants needed to build new cells and carry out many activities. These minerals included nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and several smaller amounts of other elements that were still necessary for healthy growth. The first true roots were probably much simpler than the roots of many modern plants. They were smaller and had fewer branches, but they already showed the important ability to move water and minerals upward through special tissues. Over many millions of years, roots became more complex. They formed many branches that spread through large areas of soil. This allowed plants to collect more water and nutrients, making it possible for them to grow larger than their earlier ancestors. As roots became stronger, plants were able to live in places that were not constantly covered with water. They no longer depended only on rain falling directly onto their bodies. Water collected by the roots could travel upward to the rest of the plant. This greatly increased the number of environments where plants could survive. Damp forests, riverbanks, open ground, and many other habitats slowly became home to different kinds of early plants. Inside these developing roots, special conducting tissues began to appear. One type of tissue carried water and dissolved minerals from the roots upward into the rest of the plant. Another type carried sugars made during photosynthesis from the green parts of the plant to areas where energy was needed. These conducting tissues made the movement of materials much more organized than in earlier plants, where water mostly moved from one cell directly into the next. The growth of stems was closely connected to the appearance of roots. Without roots supplying water, tall stems would not have been possible. Stems became the main support of the plant body. They lifted the green parts upward where sunlight was easier to reach. Since sunlight provides the energy needed for photosynthesis, growing upward gave many plants a better chance to produce food. The earliest stems were not thick or woody. They were soft and flexible. Their conducting tissues carried water upward from the roots while transporting food throughout the plant. Even these simple stems gave early land plants an important advantage because they allowed different parts of the plant to specialize. Roots focused mainly on collecting water and minerals. Green parts focused mainly on making food. Stems connected these different parts into one working system. As stems slowly became stronger, plants were able to grow higher above the ground. This helped them spread their reproductive structures over greater distances. Spores released from taller plants could be carried farther by the wind, increasing the chance that new plants would grow in different places. This wider spread helped many plant groups expand into new environments. The strengthening of stems happened through gradual changes in their tissues. Cells developed thicker walls that contained substances able to provide greater support. One of the most important of these substances was lignin. Lignin made cell walls harder and stronger. It helped stems remain upright while carrying the weight of the growing plant. Lignin also improved the movement of water by strengthening the conducting tissues inside the stem. The appearance of lignin marked an important stage in plant history because it allowed many plants to become much taller than before. Instead of remaining close to the ground, some plants eventually formed upright bodies that reached much greater heights. Taller plants received more sunlight and spread their spores over larger areas. As stems became stronger, branching also became more common. Instead of producing only one upright shoot, many plants formed several branches. Each branch carried additional green tissues that increased the total amount of photosynthesis. More photosynthesis meant more food, supporting even greater growth. Branching created many different shapes among early plants. Some plants developed only a few branches, while others produced large networks of branches. These patterns depended on small genetic differences that accumulated over countless generations. Natural selection favored forms that worked well in particular environments. The earliest leaves did not appear suddenly. Before true leaves evolved, photosynthesis took place mainly in the green stems. The stems themselves captured sunlight and produced sugars. As branching became more complex, some branches became flatter. These flattened structures exposed more surface area to sunlight. Over time, these flattened parts gradually developed into the earliest primitive leaves. Primitive leaves were much smaller and simpler than the leaves seen on many flowering plants today. They contained only basic tissues and were often narrow in shape. Even so, they greatly increased the amount of sunlight that plants could capture. This allowed plants to produce more food and support larger bodies. Scientists believe that different groups of plants developed leaves in different ways. Some primitive leaves formed from small outgrowths on the stem. Others developed from flattened branching systems. Although these pathways differed, they all increased the amount of photosynthetic tissue available to the plant. The earliest small leaves are often called microphyls. These leaves usually contained only a single strand of conducting tissue. They appeared in early relatives of modern club mosses. Their simple structure still provided important benefits because even a small increase in photosynthetic area helped plants produce more energy. Larger primitive leaves, known as megaphylls, developed later in another group of plants. These leaves contained many branching veins that carried water into the leaf while transporting sugars away from it. The network of veins also strengthened the leaf, allowing it to grow larger without tearing easily. The development of veins inside leaves greatly improved plant function. Water reached cells more efficiently, and sugars moved quickly to growing tissues. This increased the overall productivity of the plant and supported faster growth. As leaves expanded, plants faced another challenge. Larger surfaces increased the loss of water into the air. To reduce this problem, leaves developed a waxy covering called the cuticle. The cuticle slowed the escape of water while still allowing sunlight to reach the cells underneath. Leaves also developed tiny openings called stomata. These openings allowed carbon dioxide to enter for photosynthesis and oxygen to leave the plant. Each opening could open and close depending on environmental conditions. When water was plentiful, stomata opened more freely. During dry conditions, they closed to reduce water loss. The ability to control these openings gave plants much greater success on land. Instead of constantly losing water, they could adjust their gas exchange according to changing weather. This increased their chances of surviving periods of dryness. The growth of roots, stems, and primitive leaves did not happen separately. Each new structure supported the others. Stronger roots supplied more water. Better stems carried that water upward. Larger leaves used the water during photosynthesis to produce more food. The food then moved through the conducting tissues to support additional growth in every part of the plant. This connected system gradually became more efficient as new adaptations continued to appear over many millions of years. The rise of vascular plants marked one of the most important stages in the long history of plant life on Earth. Before vascular plants appeared, most plants were small and lived in places where water was always close by. They depended on wet surroundings because they could not move water or nutrients very far inside their bodies. This kept them close to rivers, lakes, ponds, wetlands, and damp soil. Their size was limited because every part of the plant had to remain near a source of moisture. As life continued to change over many millions of years, some plants slowly developed special tissues that could carry water, minerals, and food throughout the entire plant. This new ability completely changed the way plants lived, grew, and spread across the land. The earliest land plants had already shown that life could exist outside the oceans. They had developed ways to survive sunlight, drying air, and changing temperatures. Even so, these early plants still faced many problems. Water entered their bodies mostly through direct contact with the soil or through their entire surface. Once water entered, it could only move short distances. This meant that these plants stayed low to the ground because taller bodies would not receive enough water in their upper parts. Their leaves, stems, and other structures remained simple because they could not support large amounts of living tissue. As generations passed, small changes appeared in different groups of plants. These changes happened through natural variation. Plants that carried water more effectively often survived better in places where water was not always available. Over long periods of time, these useful features became more common. Gradually, some plants developed long cells that formed continuous tubes inside their stems and roots. These tubes became the beginning of the vascular system. The vascular system included two main kinds of tissues. One tissue carried water and dissolved minerals from the roots upward through the stem and into the leaves. This tissue became known as xylem. The second tissue carried sugars and other food made during photosynthesis from the leaves to the rest of the plant. This tissue became known as phloem. Together, these tissues formed a transport network that reached every part of the plant. Water, minerals, sugars, and other important materials could now move over much greater distances inside the plant than before. The appearance of xylem changed plant life in many ways. Water collected by the roots no longer had to move slowly from one cell to another across the entire plant body. Instead, it traveled through long tubes that stretched from the roots to the highest growing points. Minerals dissolved in the water also moved through these tubes. Cells in distant parts of the plant received the materials they needed for growth. This allowed plants to become taller, because even leaves far above the ground could receive a steady supply of water. Phloem also became an important part of plant success. Leaves use sunlight to make sugars through photosynthesis. These sugars served as food for growth, repair, storage, and reproduction. Without phloem, food could only move slowly between nearby cells. Once phloem evolved, sugars traveled throughout the plant. Roots received food even though they stayed underground without sunlight. Growing stems, developing leaves, and reproductive structures all received energy where it was needed. This made growth much more efficient. The evolution of vascular tissue also gave plants greater strength. Xylem contained thick cell walls that became hardened with a material called lignin. Lignin made these cells stiff and strong. Stronger tissues allowed stems to remain upright instead of bending toward the ground. Plants no longer depended only on water pressure inside their cells for support. Their stems became firmer and more durable. Lignin became one of the most important materials ever produced by plants. It strengthened stems, branches, and roots. Plants could now support larger bodies without collapsing under their own weight. As stems grew taller, they lifted leaves higher into the sunlight. This increased the amount of light available for photosynthesis. More sunlight often meant more food production, leading to faster growth and greater survival. The ability to grow upward created many new opportunities. Taller plants captured sunlight before shorter plants could reach it. This encouraged the evolution of even taller forms. Over many generations, forests began to appear in places where only low plants had once existed. Layers of vegetation developed, with tall plants rising above smaller ones. The structure of plant communities became more complex. Roots also changed as vascular plants evolved. Earlier land plants often had simple structures that attached them to the soil but did not function as true roots. Vascular plants gradually developed deeper and more effective root systems. These roots anchored plants securely in the ground and absorbed water and minerals from deeper layers of soil. The increased supply of water supported larger stems and more leaves. Deeper roots also helped plants survive dry periods. Water stored beneath the surface often remained available after rain had stopped. Plants with deeper roots continued to absorb moisture, while shallow-rooted plants dried out more quickly. This allowed vascular plants to spread into areas that were less consistently wet. As roots expanded through the soil, they also affected the land itself. Growing roots broke apart rocks into smaller pieces. Tiny cracks widened as roots expanded. Water entered these cracks and continued the process of breaking rocks apart. Dead roots added organic matter to the soil. Over many generations, soils became richer and deeper. These improved soils supported even more plant growth. Leaves also became more advanced during the rise of vascular plants. Early leaves were often very small or completely absent. As vascular tissues spread throughout the plant body, larger leaves became possible. Veins carrying xylem and phloem reached every part of the leaf. Water entered the leaf while sugars moved away after photosynthesis. Strong veins also supported the leaf structure. Larger leaves collected more sunlight. Greater exposure to sunlight allowed plants to produce more sugars. More sugars supported faster growth, larger stems, stronger roots, and greater reproduction. Plants with efficient leaves often produced more spores or seeds than those with smaller leaves. This increased their chances of spreading into new areas. The appearance of vascular plants also changed reproduction. Many early vascular plants still reproduced through spores. spores formed inside special structures and were released into the air. Wind carried many spores over long distances. If they landed in suitable places with enough moisture, they could grow into new plants. Producing large numbers of spores increased the chances that some would survive. Although spores remained important, vascular plants gradually became better at protecting their reproductive structures. Stronger stems held spore-producing organs above the ground where wind could spread them farther. Greater height increased the distance that spores travelled. This helped plants reach new habitats and establish larger populations. The growing bodies of vascular plants required more energy than smaller plants. Efficient transport systems made this possible. Water moved upward continuously while sugars moved to every growing tissue. Cells divided more rapidly because they received the resources they needed. Growth became faster than in earlier land plants. Faster growth brought many advantages. Plants reached maturity sooner and produced reproductive structures earlier. They repaired damaged tissues more effectively. New leaves replaced old ones as needed. Strong root systems expanded into fresh soil. Plants became better at responding to changing environmental conditions. The rise of vascular plants also affected Earth's atmosphere. Larger plants carried out photosynthesis on a much greater scale. During photosynthesis, plants removed carbon dioxide from the air and released oxygen. As forests expanded, this process became increasingly important. More carbon became stored inside wood, roots, leaves, and eventually in the soil after plants died. Large amounts of dead plant material accumulated in wetlands and forests. Some of this material decomposed slowly because oxygen levels inside wet soils remained low. Over extremely long periods, buried plant remains contributed to the formation of coal deposits. These deposits preserved carbon that had once existed in the atmosphere. The spread of vascular plants changed animal life as well. Small animals found shelter beneath stems and among leaves. Plant tissues became food for many early plant-eating animals. Dead leaves provided food for fungi and bacteria. Insects found places to feed, hide, and reproduce. As plant life became richer, animal communities also became more diverse. The appearance of taller vegetation influenced local climates. Dense plant cover shaded the soil and reduced water loss. Leaves released water vapor into the air through tiny openings called stomata. This process contributed moisture to the atmosphere. Local humidity increased in many vegetated areas. Rainfall patterns may also have been influenced by the expanding plant cover over long periods. The development of stomata represented another important improvement. Stomata allowed plants to exchange gases with the atmosphere. Carbon dioxide entered through these tiny openings, while oxygen and water vapor left the plant. Guard cells surrounding each opening controlled when the stomata opened and closed. This helped plants balance the need for carbon dioxide with the need to reduce water loss. Plants living in drier environments benefited greatly from this control. During hot or dry conditions, stomata could close to reduce the escape of water. During favorable conditions, they opened to allow photosynthesis to continue. This increased the ability of vascular plants to survive in many different climates. The earliest vascular plants were still fairly simple when viewed beside later trees and flowering plants. Many lacked large leaves or complex branching patterns. Even so, their internal transport system represented a major step forward. Every later group of large land plants inherited this basic system and built upon it. Over time, stems became more branched. Branching increased the number of leaves that could receive sunlight. More leaves meant greater food production. The extra sugars supported additional branches. creating larger and more productive plants. Each new branch also carried reproductive structures, increasing the number of spores produced. The evolution of branching also helped plants recover after damage. If one branch died, others continued growing. Resources could move through the vascular system to support healthy parts of the plant. This flexibility increased survival in changing environments. Many early vascular plants lived in warm and moist regions because these conditions still favored plant growth. However, their improved transport systems allowed them to spread into a wider variety of habitats than earlier land plants. Riverbanks, floodplains, open ground, and gradually drier, landscapes became occupied by different vascular plant species. Competition for sunlight became stronger as plants grew taller. Individuals that reached greater heights often captured more light and produced more food. Natural selection favored traits that supported upright growth, stronger stems, improved roots, and efficient transport tissues. These traits spread through populations over many generations. The growing diversity of vascular plants produced many different body forms. Some species remained small with thin stems. Others became much taller with many branches. Different leaf shapes appeared. Root systems expanded in different ways depending on soil conditions. Plant life became increasingly varied across the continents. Fungi continue to play an important role in plant success. Many vascular plant roots formed close relationships with fungi living in the soil. The fungal threads reached into tiny spaces that roots alone could not easily enter. They collected water and minerals and passed them to the plant. In return, the plant supplied sugars produced during photosynthesis. These partnerships improved plant nutrition and supported healthy growth. Bacteria also contributed to soil fertility. As dead plants decomposed, bacteria broke down organic material into simpler substances. Nutrients returned to the soil where roots absorbed them again. This recycling supported continuous plant growth across many generations. The increasing size of vascular plants influenced erosion. Dense root systems held soil together. During heavy rain, slopes became more stable because roots reduced the amount of loose soil washed away by flowing water. Rivers carried different amounts of sediment as vegetation spread across the land. Plant litter accumulated on the ground beneath vascular plants. Fallen leaves, broken branches, dead roots, and other plant remains formed a layer of organic material. This layer helped retain moisture, protected the soil from direct sunlight, and provided food for decomposers. Richer soils supported even more plant growth. As forests expanded, sunlight reaching the ground decreased beneath dense canopies. Some plants adapted to these shaded conditions, while others continued growing in open areas. Different plant communities developed depending on the amount of available light, soil moisture, temperature, and nutrients. The internal structure of vascular plants continued becoming more organized. Specialized tissues performed different jobs. Some cells focused on transport, others stored food. Some provided support while others carried out photosynthesis. This division of work increased efficiency because each tissue became well suited for its own function. Growth occurred mainly in special regions where cells divided rapidly. These growing points appeared at the tips of roots and stems. New cells formed continuously. Some became transport tissues while others developed into leaves, roots, or supportive structures. This organized growth allowed plants to increase in size throughout much of their lives. As stems thickened, additional supportive tissues developed around the vascular system. These tissues protected the transport tubes from damage. Strong outer layers also reduced water loss and defended the plant against physical injury. The spread of vascular plants influenced the movement of water across landscapes. Rain falling onto forests followed many different paths. Some water remained on leaves before dripping to the ground. Some entered the soil through root channels. Some evaporated back into the air from leaf surfaces. These processes affected streams, rivers, and groundwater over long periods. Plant roots also increased the amount of water entering the soil. Root channels created spaces where rainwater could move downward more easily. Better water movement supported underground ecosystems and maintained moisture for future plant growth. Different vascular plant groups gradually appeared as evolution continued. Some lineages gave rise to club mosses, horsetails and ferns. These groups still reproduced with spores but developed many successful forms that spread widely across ancient landscapes. Each group showed different arrangements of stems, leaves, and reproductive structures, while sharing the basic vascular system. Club mosses became common in many ancient forests. Some remained small, while others reached much larger sizes than modern relatives. Their stems contained vascular tissues that supported upright growth. Their spores formed in specialized structures near the tips of shoots. Horsetails developed hollow stems with joints and narrow leaves arranged around the stem. Their vascular tissues allowed efficient transport throughout the plant. They often grew in moist habitats where underground stems spread through the soil and produced many new shoots. Ferns became one of the most successful groups of early vascular plants. Their large divided leaves collected sunlight efficiently. Spores formed beneath mature leaves in specialized clusters. Ferns spread across many parts of the ancient world and remain common today in many forests. Although these groups differed in appearance, they all depended on vascular tissues to transport water, minerals, and sugars. Their success demonstrated the importance of internal transport systems in plant evolution. As vascular plants increased in size, their life cycles also became dominated by the larger plant body known as the sporophyte. Earlier land plants had life cycles where another stage played a more obvious role. In vascular plants, the sporophyte became stronger, larger, and longer-lived. It carried out most photosynthesis and produced spores for reproduction. This shift allowed plants to invest more energy in building large bodies with extensive vascular tissues. Strong sporophytes supported greater reproduction because they produced large numbers of spores from elevated positions. The rise of vascular plants prepared the way for later developments, including seeds. Although seeds had not yet become widespread during the earliest stages of vascular plant evolution, the transport system, supportive tissues, roots, and leaves created the foundation upon which seed plants later evolved. Faster growth remained one of the greatest advantages provided by vascular tissues. Water reached growing cells quickly. Sugars moved efficiently to every organ. Nutrients traveled where they were needed. Waste products also moved through plant tissues. Every part of the plant benefited from improved internal movement. Rapid growth increased the ability of plants to recover after storms, floods, droughts, grazing, and physical damage. New shoots developed from growing points. Damaged roots were replaced, leaves regenerated. Plants that recovered quickly often survived to reproduce successfully. Large vascular plants also stored greater amounts of food inside roots, stems, and underground structures. These reserves supported new growth during favorable seasons or after periods of stress. Stored energy increased the chances of survival during difficult conditions. The expansion of vascular plants influenced the evolution of many living organisms. Plant-eating animals adapted to consume stems, leaves, roots, and spores. Predators followed these animals into plant-rich habitats. Fungi specialized in decomposing woody tissues, containing lignin. Bacteria adapted to recycle nutrients locked inside dead plant material. The growing amount of oxygen released by expanding vegetation supported increasingly active forms of life. Carbon stored within plant tissues helped regulate atmospheric composition over very long periods. The interaction between plants, soil, water, air, fungi, bacteria, and animals became increasingly connected. As generations continued, natural selection kept improving vascular systems. Plants with wider transport tissues often moved water more efficiently. Better root systems increased mineral absorption. Improved leaves raised photosynthesis rates. Stronger stems supported larger bodies. Small changes accumulated slowly across countless generations. Different environments favored different adaptations. Plants living in wet habitats often developed features unlike those growing in dry regions. Mountain plants faced different conditions from lowland species. Even within forests, plants growing under shade developed differently from those exposed to full sunlight. The vascular system remained essential in every habitat, but each species adjusted its growth to local conditions. Seasonal changes also influenced vascular plant growth. During favorable periods with enough water and sunlight, transport tissues supplied growing cells continuously. During less favorable times, growth slowed while stored food helped maintain living tissues. When conditions improved, growth resumed using both stored reserves and newly produced sugars. The development of bark in later vascular plants provided additional protection. Outer tissues shielded the living vascular system from injury, drying, and harmful organisms. This allowed many plants to survive for much longer periods while continuing to grow year after year. Wood gradually became one of the defining features of many vascular plants. Layers of xylem accumulated inside stems and branches. Each new layer strengthened the plant while improving water transport. Larger trunks supported extensive branches, carrying thousands of leaves. This increased photosynthesis and allowed even faster overall growth. The ability to produce wood also increased lifespan. Instead of completing their life cycle quickly, many vascular plants survived for many years. Longer lives meant repeated opportunities for reproduction. Mature plants often produced far more spores or seeds than young plants. The spread of vascular plants transformed the appearance of Earth's continents. Areas once covered mainly by low vegetation became filled with taller plants, dense forests, and rich soils. Rivers flowed through wooded landscapes. Wetlands became surrounded by extensive vegetation. New habitats appeared for countless forms of life. Every improvement in transport, support, rooting, leaf structure, and growth allowed plants to occupy places that had once been difficult or impossible to survive in. Their expanding presence continued shaping Earth's atmosphere, soils, waterways, and living communities while providing the biological foundation for the later evolution of forests, seed plants, flowering plants, and the rich ecosystems that developed across the continents. Plant reproduction has always been one of the most important parts of the history of plants, because every generation of plants depends on the successful production of new life. From the earliest plant-like organisms living in water to the first seed-producing plants growing across the land, reproduction slowly changed over a very long period of Earth's history. These changes allowed plants to spread into new environments, survive difficult conditions, and become more successful. Every new method of reproduction gave plants new ways to continue their existence, even when conditions were not always favorable. The story of plant reproduction began with very simple methods and slowly became more advanced as plants adapted to life on land. The earliest plant-like organisms lived in water, where reproduction was much easier because water helped move reproductive cells from one place to another. Many simple algae reproduced by dividing into new cells. One cell became two cells, and each new cell continued growing as a separate organism. This method allowed large numbers of new organisms to appear quickly whenever enough sunlight, water, and nutrients were available. Some algae also reproduced by breaking into smaller pieces. Each piece continued growing into a complete organism. This type of reproduction did not require special reproductive structures because each fragment already contained living cells that could continue growing. As algae became more diverse, many species developed special reproductive cells. Some produced cells that could swim through water using tiny structures called flagella. These moving cells searched for other reproductive cells so they could join together. When two reproductive cells joined, they formed a new cell that contained genetic material from both parents. This mixing of genetic material created new combinations of inherited traits. These new combinations helped populations survive changing conditions because not every individual was exactly the same. Water played an essential role in the reproduction of these early organisms. Without water, these swimming reproductive cells could not move toward each other. Rain, rivers, ponds, lakes, and shallow seas provided the environment needed for successful reproduction. Since water surrounded these organisms, drying out was usually not a major problem. The reproductive cells remained protected by the surrounding water until new individuals began growing. As the ancestors of land plants slowly evolved, they faced many new challenges. Life on land offered more sunlight and more open space, but it also brought serious problems. Dry air could damage delicate reproductive cells. Strong sunlight could harm exposed tissues. Wind could carry tiny cells away before reproduction was complete. Plants needed new ways to protect their reproductive processes while still producing enough offspring to survive. One of the earliest and most important developments in land plant reproduction was the production of spores. Spores are tiny reproductive cells that can grow into new plants without joining with another spore. Each spore contains everything needed to begin a new stage of growth when conditions become favorable. Spores are very small, making them easy for wind to carry over long distances. Some also move through flowing water or are carried by animals. Spores developed strong outer walls that protected them from drying out. These walls allowed spores to survive much longer than delicate reproductive cells that lacked protection. Even when conditions became dry or cold, many spores remained alive until enough moisture returned. This ability greatly increased the chances of survival. A spore could remain inactive for a period before beginning to grow when the environment became suitable again. The protective wall around a spore contains durable materials that resist damage from sunlight, drying, and physical stress. Because of this protection, spores became one of the most successful reproductive methods in the plant kingdom. Even today, many plants continue using spores as their main method of reproduction. Mosses, liverworts, hornworts, ferns, club mosses, and horsetails all produce spores. The earliest land plants likely depended almost entirely on spores for reproduction. These small plants lived close to moist ground because they still required water for other stages of their reproductive cycle. Although spores could survive dry conditions, another important step still depended on water. After spores grew into small plants, these plants produced reproductive cells that needed moisture to reach one another. The life cycle of these early plants became more complex than the life cycles of many algae. Instead of growing directly from one generation into another identical generation, plants developed two separate stages that alternated throughout their lives. This process is called the alternation of generations. One generation produced spores, while the other produced reproductive cells. Both generations played important roles in completing the life cycle. The stage that produces spores is known as the sporophyte. This generation develops special structures where spores are formed. Inside these structures, cells divide and eventually produce many spores. When the spores become fully developed, they are released into the environment. Wind often carries them away from the parent plant. Some land nearby, while others travel much greater distances before reaching suitable places for growth. When a spore lands where enough moisture, light, and nutrients are available, it begins to grow. Instead of becoming another sporophyte immediately, it develops into a different generation called the gametophyte. This stage is usually much smaller in early plants. The gametophyte produces reproductive cells known as sperm and eggs. These reproductive cells eventually unite to begin another sporophyte generation. The sperm cells produced by early land plants still required water to swim toward the eggs. Rainwater often formed a thin layer across the ground, allowing the sperm to move. Without enough water, fertilization could not take place. Because of this requirement, many early land plants remained closely connected to damp environments, even though they had already adapted to living on land. Mosses provide one example of plants that still show many features of these ancient reproductive systems. The green moss plants, commonly seen today, are actually the gametophyte generation. They produce sperm and eggs in separate reproductive organs. When water is available, sperm swim to the eggs and fertilization occurs. The resulting young sporophyte grows directly from the gametophyte and remains attached to it during development. The sporophyte of a moss eventually produces a capsule at its tip. Inside this capsule, many spores develop. As the spores mature, the capsule opens, allowing the spores to escape into the air. Wind carries them away to begin new life cycles. A single capsule may produce thousands of spores, increasing the chances that some will land in suitable places. Liverworts also reproduce using spores. Many liverworts grow close to the soil in damp forests, wetlands, and shaded places. Like mosses, they require water for fertilization. Their reproductive structures differ in shape, but the basic process remains similar. Spores develop inside protective structures and are released when mature. Hornworts follow a similar reproductive pattern. Their long, narrow spore-producing structures continue growing for extended periods, releasing spores gradually instead of all at once. This longer release period increases the chances that at least some spores will find favorable conditions. As plants continued evolving, vascular tissue appeared. Vascular tissue allowed water and nutrients to move efficiently throughout the plant body. With this improvement, plants grew taller and spread farther across the land. Their reproductive systems also became more successful because taller plants released spores from greater heights. Wind could carry these spores much farther than before. Ferns became one of the most successful groups of spore-producing vascular plants. They spread across forests, wetlands, and many other environments. Fern leaves produce clusters of spore-producing structures, usually found on the undersides of mature leaves. Each cluster contains many small spore cases. When the spores mature, the cases open and release them into the air. A fern spore grows into a tiny heart-shaped gametophyte. This small plant lives independently and produces both sperm and eggs. Since it remains very small and close to the ground, it still depends on moisture for fertilization. Once fertilization occurs, a young fern sporophyte begins growing from the gametophyte. As it becomes larger, it develops roots, stems and leaves, while the small gametophyte eventually dies. Club mosses and horsetails also reproduce through spores. Although they differ in appearance from ferns, they share many similar reproductive steps. Spores develop in special structures, are released into the environment, grow into gametophytes, and eventually produce new sporophytes after fertilization. Producing many spores gave plants important advantages. A single plant could release enormous numbers of spores during one growing season. Even if only a small percentage survived, enough new plants could still become established. Wind carried spores across hills, valleys, rivers, and open plains. This wide distribution helped plants spread into new regions. The small size of spores also allowed them to enter tiny spaces where larger reproductive structures could not fit. They settled into cracks in rocks, thin layers of soil, and other protected places where moisture collected. These small spaces often provided safe environments for early growth. Although spores offered many advantages, they also had important limits. Most spores contained only a small supply of stored food. After germination, the young plant needed favorable conditions almost immediately. If moisture disappeared or nutrients were lacking, the young plant often died before reaching maturity. Another challenge involved fertilization. Even though spores themselves could survive dry conditions, sperm still required water to reach eggs. During long dry periods, reproduction stopped until rainfall returned. Plants could survive, but they often could not produce new generations until moisture became available again. Over millions of years, natural selection favored plants that found ways to reduce their dependence on standing water during reproduction. Small changes accumulated gradually across many generations. Some plants developed new reproductive structures that protected developing offspring more effectively. Others produced different types of spores with specialized functions. One important development was the appearance of two different kinds of spores instead of only one. Some plants began producing very small spores and much larger spores. This condition is called heterospory. The smaller spores developed into male gametophytes, while the larger spores developed into female gametophytes. The larger spores contained more stored food, giving the developing female gametophyte greater support during early growth. This additional nourishment increased the chances of successful development. The female gametophyte often remained protected inside the parent plant instead of becoming completely independent. The smaller male spores became specialized for delivering sperm. Because they were lightweight, wind could carry them over long distances. This separation of male and female functions marked an important step towards the evolution of seeds. Several ancient groups of vascular plants independently developed heterospory. Although not all survived into the present day, they demonstrated important reproductive innovations. These changes gradually reduced the risks associated with reproduction on dry land. Inside the larger spore, the female gametophyte developed while remaining enclosed within protective tissues. The egg stayed safely inside these tissues instead of being fully exposed to the outside environment. This protection greatly increased the survival of developing reproductive structures. At the same time, the male reproductive structures became increasingly specialized. Rather than releasing swimming sperm directly into open water, plants gradually developed ways to transport male reproductive cells closer to the female structures before fertilization occurred. Another major step involved the evolution of pollen. Pollen grains are tiny structures that contain the male reproductive cells. Unlike swimming sperm released directly into water, pollen can travel through the air. Wind became one of the main methods of carrying pollen from one plant to another. The outer wall of pollen provides strong protection against drying. This protective covering allows pollen to survive during travel through the air. Some pollen grains remain alive for several days before reaching suitable female structures. Others survive even longer under favorable conditions. When pollen reaches the female reproductive structure, it begins growing a pollen tube. This tube carries the male reproductive cells toward the egg without requiring freestanding water. This development greatly reduced the plant's dependence on rainfall during fertilization. The appearance of pollen represented one of the most important events in plant evolution because it allowed reproduction to continue even in much drier environments. Plants no longer needed a continuous layer of water for sperm movement. Fertilization could occur inside protected tissues. While pollen solved one major problem, plants also developed another remarkable structure that protected the next generation. This structure became known as the seed. A seed begins forming after fertilization. The fertilized egg develops into an embryo, which is the young plant. Around the embryo, protective tissues form a seed coat. Food reserves also develop inside the seed to nourish the young plant during its early growth. The seed coat protects the embryo from drying, disease, physical injury, and changing weather. Unlike spores, seeds contain a much larger food supply. This stored food allows the young plant to continue growing even if environmental conditions are not immediately perfect. The embryo inside a seed remains alive but inactive until suitable conditions arrive. This resting stage is called dormancy. During dormancy, the seed can survive cold weather, dry periods, or other unfavorable conditions. When moisture, oxygen, and suitable temperatures return, growth begins. The first seed-producing plants appeared during the late Paleozoic era. These early seed plants still differed greatly from many modern flowering plants, but they introduced a reproductive system that became extremely successful across the world. Ancient seed ferns were among the earliest seed-producing plants. Although their leaves looked similar to fern leaves, they reproduced with seeds rather than spores. These plants became widespread across ancient forests and formed an important part of many ecosystems. Seed ferns no longer depended on freestanding water for fertilization because pollen carried the male reproductive cells. The female reproductive structures remained protected inside specialized organs. After fertilization, seeds developed and eventually separated from the parent plant. The earliest seeds were relatively simple, but they already contained the essential parts found in modern seeds. Each seed included an embryo, stored food, and a protective outer covering. These basic features have remained successful throughout plant evolution. As seed plants became more common, they expanded into environments that had been difficult for spore-producing plants to occupy. Drier landscapes supported growing populations of seed plants because reproduction no longer depended so heavily on rainfall during fertilization. Wind remained an important method of transporting pollen. Some early seed plants produced enormous amounts of pollen to increase the chances that some grains would reach female structures. This strategy continues among many living gymnosperms today. The evolution of seeds also changed how young plants began life. Instead of depending almost entirely on immediate environmental conditions after germination, seedlings received nourishment from food stored inside the seed. This support increased survival during the earliest stages of growth. Some seeds remained dormant for extended periods before germinating. This delayed growth allowed plants to avoid beginning development during unfavorable seasons. Seeds often waited until enough moisture and suitable temperatures became available. The protective seed coat also reduced damage from tiny animals, fungi, bacteria, and physical injury. Although many seeds were still eaten by animals, enough survived to continue future generations. Early seed plants gradually diversified into many different groups. Some eventually gave rise to cycads, ginkgo relatives, conifers, and other gymnosperms. Much later, flowering plants appeared and developed even more advanced reproductive systems involving flowers, fruits, and animal pollination. Despite these later developments, the basic reproductive steps established by the earliest seed plants remained largely unchanged. Pollen carried male reproductive cells. Fertilization occurred within protected tissues. Embryos developed inside seeds. Food reserves supported early growth. Protective seed coats guarded the young plants until favorable conditions returned. Spore-producing plants never disappeared. Mosses, liverworts, hornworts, ferns, club mosses, and horsetails continued reproducing successfully through spores. These plants still occupy forests, wetlands, mountains, grasslands, deserts, and many other environments throughout the world. Their long history demonstrates that spores remain an effective reproductive strategy under suitable conditions.